don't have. Here's my options. Where are the view options on here, man? Let's see. Okay, let's do that. Oh, that might actually work. Awesome. Good morning. Father, Son, Holy Spirit, amen. God, grant me the serenity to accept the things I cannot change, courage to change the things I can, and the wisdom to know the difference. Father, Son, Holy Spirit, amen. All right. Let's try to share screen. Okay, believe it or not, I actually did go back and look at my PowerPoint presentation, and this is not the picture. It's not even anywhere. I don't even know where that thing's coming from. It's not even on the screen, but there we have it. I don't know why it shows up on there, but I have no clue. Okay. Let's see. We got Jasmine, Callum. Okay, I'm starting to get this down. Aiden. We got Matt. Emily. Okay, Emily. Abigail. Ashley. Anna. Donovan. John. Quattro. It's Quattro. Okay. Cameron. Rebecca. John. Got it. Kate. Yo. Sophia. Maria. Oh, did you move? You did it on purpose. Don't do that. Okay, Sam. I'm just joking. Sit where you're on. Sam, over there. Okay. Christina. Okay. Got you, Christina. Kristen. Sarah. Hi, Sarah. Tristan. Okay. Caleb. Gotcha, Caleb. Kyle. Jordan. That's right, there's Jordan back there. Antonia. Okay. Deshaun. Gotcha. And Jonah. Landon. Man. And Anthony. Okay, is this working? Okay. All right. Come on. All right. So 4 levels of structure. I'm going to move these people over here, maybe. Move them right there, just barely off the good. 4 levels of structure. We still have things going on down here. Okay. Primary structure, right? The ribosomes are making the protein. They're putting the amino acids together. Bum, 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 bum. And a line. That's the primary structure. Once they go in, they start change shape. Because we start to get what kind of bonds form? Anna? We get hydrogen bonds. All right, hydrogen bonds begin to form. Like here is a possible place for a hydrogen bond here between amino acids. So for secondary structure, the R group is or is not involved? Is not. So you should be able to, if you saw something like this, it'd be a good practice for you to be able to identify individual amino acids where we would find them. Okay. Anyways, this is another example of a hydrogen bond. The little dots here. This is an example of a hydrogen bond. All right. So this hydrogen and this oxygen are not from an R group. Now it's important that I'm highlighting this here because we are going to see that there's the next level of structure, which is tertiary structure, which involves the R group. It's possible, and oftentimes happens, that hydrogen bonds that will look very similar to this will form between R groups. So if a hydrogen bond forms that helps bring about secondary structure, it's not involving the R group. Now, in general, these secondary structures have 2 major shapes. One is what's called a beta sheet. Should probably put alpha helix first. So alpha helix. All right, so the beta sheet looks, if you were to look at it as a fully formed protein, it looks kind of like it. They usually represent it as ribbons. Kind of looks like ribbons, huh? They look like ribbons. Now, an example of a beta sheet is what's called a pore, P-O-R-E, protein. And a pore, now this structure here, this is a plasma membrane. So I hope maybe you recognize this kind of structure. So this is a plasma membrane. And this beta sheet, or this protein, is forming a pore protein. And it's literally, it's a protein hole that goes right through the plasma membrane. That's what a pore protein is. All right, you saw that. So, an example of something that's comprised of alpha helices would be not a lion, although lions are, hair, right? That's an example of something that sort of has that shape. A helix, alpha helix. That's a one helix. Looks like a spring, okay? So hair has this structure. Alpha helix. So these are just some examples. Now it's time to talk about the third level of structure. So protein is needed. Dun, dun, dun. Protein is needed. We transcribe the DNA. We make a message RNA. Gets exported. Then that begins to be translated by the ribosome. Boom, boom, boom. We put the amino acids together in sequence, forming what? Did you guys have this? No. Forming what kind of bonds? What's the bond? What's the bond between amino acids? Donovan? It is a covalent bond, specific kind of name. Anyone? Okay, John. Peptide, right? Peptide, okay. As opposed to riptide. Yo, hey, that's a new one. Boom, boom. All right. So we form these strings, these long strands. They begin to form secondary structures, hydrogen bonds between carbonyls and aminos. Form two primary shapes, alpha helix or beta sheet. And then once those begin to form, then those secondary structures begin to fold in on one another. Okay? And then we start getting tertiary structure. And this is the final folded shape of a protein. So that's the last step in that protein or that peptide becoming what it's supposed to be. Kind of. And we're going to see what I mean by kind of in a moment. But this is the final folded shape. And the tertiary structure results from interactions between R groups. So here we could see, perhaps at this loop, there might be, although it's not being represented, there may be an interaction between these two R groups that helps contribute to that loop. Okay. Now there are 4 different kinds of interactions between R groups that contribute to the tertiary structure of a peptide or protein. So we'll go through these one by one. Uno by uno. Un by un, as they say en français. First one, hydrophobic exclusion. Hydrophobic exclusion. So, remember, these are R groups. These are R groups. So which major category of amino acids is comprised of amino acids with R groups that would form hydrophobic, that are hydrophobic. 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 Okay, Aiden? Non-polar. Non-polar molecules do not interact with water very well, ergo they are hydrophobic. And they are primarily made up of which two elements? Jacob. Jordan, sorry. Jordan. They're made up of which two elements mainly? Hydrophobic. Good, right? Hydrocarbons. That's what we see here. So, does anyone ever put water on oil? No. Oil on water? No. Water on oil? Doesn't matter. Doesn't matter. Do they mix? No. What does the oil do? What's it do if you put it in water? Okay. It separates. Okay. Sometimes it just forms a big layer, right? But they don't mix, right? They don't mix. So you might even see like maybe you're talking about, Cameron, is like little globules of oil, right? Floating in the water. So the hydrocarbons, they come together, they literally push out the water. They push it out. Hydrophobic, they don't like water. Exclusion, push it out. Like a squeegee. Anyone know what a squeegee is? Squeegee. So it's a molecular, cellular squeegee. Eek! Get the water out. That's what it does. All right, so nonpolar R groups. They come together to push the water away, push it out. And that's the list of the amino acids. Don't bother writing all those down. We're going to post that. So don't spend the next 5 minutes writing all those down. That's from your list. You should be able to complete that. Next, hydrogen bonds. So let's assume, well, we can't assume, so we're going to block that out. Yeah, let's assume that this is, anyways. What kind of amino acids would form hydrogen bonds? Hydrogen bonds. Anna. Polar uncharged. Yo. That's right. Polar uncharged R groups form hydrogen bonds. Maybe here, this is kind of a bad example. That shouldn't be with carbon, but maybe here we're forming one. I don't know. So take a look at those. Well, this is a smaller list. Serine, threonine, asparagine, glutamine, tyrosine. These 5 can interact with each other and form hydrogen bonds. We're going to do a worksheet on Wednesday, taking us through some of these processes. So we're doing theory today. We'll do practice on Wednesday, all right? Cool. Ionic bonds. We have a clue up here as to which class of amino acids would interact to form ionic bonds. Kristen. Phosphate, okay. Well, that's a functional group. So we're thinking about categories of phosphate would certainly contribute to forming an ionic bond, perhaps. But what about the different categories of amino acids? If you flip back and look at your chart. Yes, the charged ones. Those that have a positive charge and those that have a negative charge. Here we're going to have an ionic bond between oppositely charged R groups. All right, and sometimes this is called a salt bridge. I'm not going to ask you about that, but these are the 5 amino acids that would contribute to forming ionic bonds. Could hold neighboring regions of the protein together to help for that final folded structure. And then finally, we've got this. We have covalent bonds can actually form. And if we have covalent bonds, those are going to be formed between sulfhydryl groups on 2 neighboring cysteines. Okay? All right. So secondary structure is the result of the formation of hydrogen bonds, period. Tertiary structure results from the formation of all different kinds of bonds and interactions between R groups. Okay. So, the first 3 levels of protein structure. If we're thinking about our grammar example again, the order of letters to form words, that could be our primary structure. Words can form a sentence, that could be secondary structure. And sentences forming paragraphs, that could be tertiary structure. Whew. Sorry, folks, my health's a little, I gotta take a break here for a minute. Sorry. All right. Okay, so the main distinction then between secondary and tertiary structure, easy way to remember. If you're trying to identify secondary structure, is always just going to be hydrogen bonds only. Secondary structure, hydrogen bonds only. Tertiary structure, all the different kinds of bonds, including hydrophobic interaction. So once all of those primary, secondary, tertiary structure, once those levels are all completed, then that particular protein or that particular, maybe it's a subunit, we're going to talk about that very shortly, it's in its final form. Now, remember going back a couple weeks ago, wow, this is week 3, right? So we go back almost to the beginning of this section. We talked about a protein called hemoglobin. Anyone remember that one? Hemoglobin, right? We said that hemoglobin is the combination of 2 alpha subunits and 2 beta subunits. So each of those alpha subunits and each of those beta subunit could be like just sort of like a completed block that I now have to combine with other blocks to form the final protein. Okay. Well, once we get to that, once we pull those 4 subunits together and we finally form hemoglobin, that's the 4th level of structure we call quaternary structure. Just a fancy way for saying 4th level of structure. So, the alpha subunit. Skir. That's the primary amino acid sequence, remember. So here's the messenger RNA coming out into the cytoplasm. Ribosomes get a hold of it, start processing, start putting together amino acids. In this order. Bum, 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 da-da-da, all the way down till it's done. There may be some there's going to be some secondary structure that forms. So we start forming some hydrogen bonds between carbonyls and aminos. Could be beta sheets, could be alpha helix. And this would be an example right here. This would be an example of tertiary structure right here. So there could be some R groups holding, or some, bonds between R groups holding some of these other alpha helices and beta sheets together. And here we have an alpha subunit. So that alpha subunit has primary, secondary, and tertiary structure. This is the final folded form of the alpha subunit. And by the way, what kinds of secondary structure do we see in here? Antonia. Hydrogen bonding, that's certainly, so hydrogen bonding is contributing to the shape of this secondary structure. What shape does that look like right there? Does it look more like a coil, or does it look more like something like ribbons wrapped on itself? It looks like a coil. So if it looks like a coil, it's an alpha helix. If it looks like a coil, so if it looks like a coil, it's an alpha helix, right? If it looks like a coil, it's an alpha, or a spring, alpha helix. So we're definitely going to go over this on Wednesday. So this is the 4th level of structure then. We bring 2 or more peptide chains known as subunits. So this is a peptide chain, peptide chain, peptide chain, peptide chain. We're going to bring 2 or more of these together to form a functional protein. And we like to use amino acids. We like to use hemoglobin because it's a great model for us, because we know what hemoglobin does, right? Carries oxygen in our bodies, right? So this is hemoglobin. This is quaternary structure. And In the beta subunit, if you get only one amino acid that switches from a valine to glutamic acid, one amino acid switches, one little mutation in the DNA. It's called a single nucleotide polymorphism, SNP. We switch from valine to glutamic acid. The beta subunit becomes sticky. And the hemoglobin then begins to clump together inside the red blood cell. And it actually changes the shape of the red blood cell. It looks almost like a crescent shape. That's why it's called sickle cell anemia. Very painful. These red blood cells can get clogged and blocked in the small arteries, cause a tremendous amount of pain. And anemia. You hear the word anemia, what does that mean? Anemia. Anna. Low oxygen level. You're not getting a lot of oxygen to your cells. That's correct. If we took a look at, let's go back and take a look at your amino acid chart. Well, I guess I could flip back using this thing. No, I don't have it on here. You could take a look and look at your amino acids and see why would that switch from a valine to a glutamic acid? What's going on there? What's happening there? Switching from a what to a what kind of amino acid? Yeah, John. We're going from non-polar to polar. So now in this one particular place in the protein, we switch from one that's maybe functioning to exclude water and combine with other amino acids to also exclude water in a region. Now, all of a sudden, it can form a hydrogen bond, maybe with different amino acids, pulling it out of its shape, okay? Awesome. Okay, cool. All right. 7 functions of proteins. You definitely need to know some of these. Enzyme catalysis. Catalyst makes reactions go faster. Enzyme, protein that makes reactions go faster. We're definitely going to talk about enzymes later. But enzymes make the reactions go faster. Faster. Always reminds me of riding on this one amusement park ride when I was a kid. We'd go into Midway. They put up those rides and you just don't really know if they're actually going to stay together, but you want to go on. There's something about you that says, I should not get on this thing because I just put it up yesterday, but I'm going to. Why? Because you're 12 years old and man, why not, right? So you spend your money, 50 cents, whatever it was back then in my day. And you'd go for a while and they'd say, does anyone on here want to go a little bit faster? And all the kids would go, yay! Anyways, enzyme catalysis. Yo. I'm here all week, folks. All right. Defense, immunoglobulins. Immunoglobulins, part of your immune response. Immunoglobulins have another name. Antibodies. Hey, we're all looking for antibody response now, are we not? Yes. And toxins. Toxins. Pufferfish. Anyone ever heard pufferfish? It'll kill you. That's a protein. Thank you very much. Ding, ding, ding. All right. Transport. Circulating transporter. So, Hemoglobin transports oxygen. All right. Anyone heard of cholesterol? Cholesterol in here. Anyone? Cholesterol. Nobody's heard of cholesterol? All right. Well, they're also transporters that transport fatty acids in your blood. And then we have membrane transporters. These are machines that literally grab a molecule and drag it from one side of the membrane to the other, either in or out. You can give me an example of a membrane transporter you've probably heard about, maybe, perhaps in a biology class at some point in your life. Sodium, potassium, ATPase, anyone? Anyone? Bueller, anyone? Anyone? Okay. Sweet. Support proteins. Cytoskeleton, cartilage. Cartilage is a glycoprotein. Your ears, they have proteins, and they've also got a little bit of sugar in there. So if you're feeling hungry, just rip a piece off, and no, you don't want to do that, okay? So cytoskeleton and cartilage. Cytoskeleton. Motion. Actin and myosin. We call that thin and thick filaments that interact in your skeletal muscle. So that way, when you go to the gym and you get all swole up, whatever they call that. They still say that, getting swole? They get all swole, man. That was like a couple years ago. It's hard to keep up with the lingo, and I don't really bother. But anyways, so motion, yes, to help you move. Regulation, hormones. A lot of hormones are proteins. We've talked about one already in this class. What was that hormone? It begins with in and ends with in. Insulin, yo. There's also proteins that regulate gene expression. And then storage. I guess. Ion binding, sure. We don't really store too much energy as protein because our proteins have work to do. But it's still a source of energy storage. All right. 7 functions of proteins. Ding, 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 ding. For this exam, you need to know at least 2 of them, right? Okay. So, I just talked about proteins. So in general, what's the name of the polymer? The polymer for proteins is? That's a monomer. Monomer is one. Polymer, Rebecca? Polymer. What's that? Yeah, it's protein. Protein is protein, yep. And then Kyle gave us the monomer, amino acids, right? We put amino acids together, forming peptide bonds, form these chains. Okay. Oh. Okay. Go ahead, Kate. What's the name of the bond that holds amino acids together? Yep. Okay. And then 2 functions. Well, I just gave you 7, so fill in your list, all right? You can do that. See the list of 7 functions of proteins. Okay, that's proteins. Now let's talk about, so we moved from the ribeye steak, at least the red part. Yum, yum, yum. Now we're going to shift to the baked potato. Everybody do the baked potato. I don't know, is that something? Do the baked potato? All right. Carbohydrates. Carbohydrates are molecules that are made of carbon, hydrogen, oxygen in a ratio of 1 to 2 to 1. Carbon, hydrogen, oxygen, 1 to 2 to 1. You need to know that. You need to know that. You need to know that. So if there are 3 carbons, how many hydrogens are there, Donovan? How many oxygens are there? 4. Got it. 1 to 2 to 1. And they taste sweet. So we call them sugars. Okay. So the monomer are called monosaccharide. Saccharide. Anyone heard of saccharin? Saccharin, anyone? Saccharin, did you ever try it? It is like sweet beyond belief. Super sweet. Way sweeter than sugar. So mono means 1, saccharide simply means sugar. So 1 sugar. We're going to talk about primarily these structures with the 5 carbons and the structures with the 6 carbons. All right, so if it has 3 carbons, 3 to 6 to 3, does everyone see that? 3 to 6 to 3. We call it a triose. Triose. So ose basically is about how many carbons you have. Ose is like carbony things. And then tri is going to be the number. So if it has, and it's important, this is really important for you to be able to, you need to count the carbons to be able to tell what kind of carbohydrate it is. So 5 carbon sugars. 1. See, now this is another thing I want to, like nomenclature. When you look at a molecule, if you don't see any letter, like see where we've got these 4 bonds and there's nothing else there, there's no letter there, assume it's a carbon. See where the number 1 is? That's a carbon right there. But there's no C. I'm sorry, that's how they write things. So there's no letter here, but that's a carbon. So 1, 2, 3, 4. Oh, the little hider, 5. We got this hiding up here. 5 carbons, those are called pentoses. Now these sugars are also, you can find them in your book. It shows you, it represents these sugars in 2 different forms. One is as a long chain, right? And the other is as a ring. I'm only going to, except for the triose, I'm only going to represent these 2 groups as rings. So you need to be able to identify them. All right, if it has 6 carbons, it is hexose. Hexose. Now, what do you notice? What's the similarity between fructose and ribose up here? What's the similarity between fructose down here and ribose up here? It's not that hard. So let's call on Sophia. What do you see similar? They do. Does the ring shape look almost like the same? So the ring shape looks almost the same. So you can't just simply look at the ring and know what kind of sugar it is. You have to count the carbons. You have to count the carbons. So let's count the carbons on our little fructose. See this one down here? This has got 2 sneaky ones. Look, there's one here. 1. 2, 3, 4, 5, 6. See that? All right, so fructose is a hexose, but it has a pentose-looking ring. It has a 5-membered ring. So you got to count the carbons. Don't simply look at the ring and say, duh, pentose. Because if you don't identify that as a hexose, you're going to be sticking fructose in your DNA. Your DNA is not going to work. Don't do that. All right. So polymers. Two sugars. So we're going to talk about different kinds of polymers. The first kind of polymer we're going to talk about are called disaccharides. Two sugars bound together. And in many other organisms, glucose is transported, by the way, here's glucose. Glucose and fructose, that makes sucrose. How I love you, how I love you. Gotta love that sucrose. Okay, so, and by the way, glucose really isn't that sweet, just in case you're wondering. On its own, not really. But anyways, so glucose is typically transported attached to another sugar to keep it from being broken down before it gets to where it's needed. We don't have that problem. But in other organisms, glucose is transported bound to another sugar. So let's look at some common disaccharides. Alpha glucose and fructose equals sucrose. That's table sugar. Love it. Notice what's happening here. What reaction is this? Deshaun, what reaction is this right here where water's coming off? We're getting rid of water. So hydrophobic isn't really forming a covalent bond. So there's a kind of bond, there's a kind of reaction that forms a covalent bond, kicks water out. Yep, what? Quattro? Dehydration, okay? So remember that. So you see that, oh yeah, that's dehydration reaction. We're forming sucrose. Glucose and glucose equals maltose. We don't talk about maltose in this class, but that's from grain. Malt, actually malted milk duds. Anyone eat milk duds? Thank you. There's someone over there that eats milk duds. Thank you very much. Thank you. That's Emily. Emily eating milk duds. Good. They're really only good because they're coated in chocolate, right? Otherwise, it's kind of like chocolate-covered powder balls. But anyways, yeah. Malt is also would be the sugar that's used. You had malt that gets fermented. We get beer. Okay, so that's the kind of sugar that yeast is using to make beer. And then we've got galactose and glucose forming lactose. That is in milk. And why do some people get, why are they lactose intolerant? Yep. So some people don't have the enzyme to break this bond, which by the way is called a glycosidic bond. To break this bond, so this thing doesn't get broken down. It doesn't get absorbed in your small intestine. And then it keeps on trucking. And when you have a lot of this molecule, you have a large number of these molecules, look at all of these hydroxyl groups. What do you think what kind of bonds can they form? All those hydroxyl groups. Landon. Hydroxyl groups. What? Hydrogen bonds, particularly with water. They pull a bunch of water with them into your intestines. Blammo. That's what happens. That's not the scientific word for it. Blammo. But anyways, you get the picture. So, cool. All right. Remember, we are going to, don't forget the quiz is up. Is it working? Good. We're going to finish with the top of page 4. Answer all those questions, the ones associated with nucleic acids and lipids. And I'm going to post a worksheet on Schoology. I'm going to well, if you want to work on it at your desk, which we're going to do on Wednesday, print it out and bring it with you on Wednesday, okay? Cool. Christina, how are you?
I'm good. I took the quiz yesterday. I'm like,
Oh, wait.